Chapter 14, the brain and the cranial nerves. These are the outcomes. We will get into the details of the brain, regions, spaces inside ventricles, the different cranial nerves, anatomical and functional classifications, and the function of each part of the brain. And generally speaking, the brain is the largest part, obviously, of the central nervous system. Remember, we did the spinal cord, which is the other part. And the brain consists of cerebrum, cerebellum, diencephalon, brainstem. We will get into the details of each part, but cerebrum, cerebellum, diencephalon, brainstem. Cerebrum, cerebellum, diencephalon, brainstem. Cerebrum is this big two hemispheres. Cerebellum, diencephalon, and brainstem are smaller parts, but we will get into the details. So here is the first part, or these are all the parts here. Cerebrum is this big, the largest one. And there is a cover outside. We call this cover cortex. And there is an inside filling, which is white matter. We will get into the details, but what you see here, this big one here, this is the cerebrum. This blue one in the back here is the cerebellum, the tiny brain. And then within this cerebrum, there is a diencephalon, and the brainstem is this part right here. Starting from here, this, and this. These parts right here. This is called the brainstem. So cerebrum, cerebellum, diencephalon, and brainstem. Cerebrum is the high mental function. Others are less. If you guys still remember when I started, I said that the whole central nervous system is managing the body. But the spinal cord is more like a supervisor, a low-level management. And then you go up to the next level, like the brainstem, the diencephalon, the cerebellum, and then the higher is the cerebrum. The highest of all is the cortex of that cerebrum. The cover that's outside here, this is your awareness, this is your consciousness, this is the highest of all. So we go in that sequence. So we start with the cerebrum, spinal cord. What's above the spinal cord? Those three. What above those three? Cerebrum. This is what we're talking about. The highest of which, the cortex. So this cerebrum is doing the highest functions. Highest functions means basically thoughts, intellectual functions, memory, decision making, planning, all these high function. It's not like you touch something and you move away. It's not something like your viscera are moving or something like that. These are all basics. But for this one, this is the highest function. There are two hemispheres, half spherical structures, and there is one right and one left. The cover of that cerebrum is called the cerebral cortex, and this is gray matter. The filling inside is white matter. If you guys are following and you have an exam coming tomorrow, you should remember that the spinal cord is gray inside and white outside. Do you remember that? The gray inside with all these horns and it's white outside. This is the opposite. The cortex outside is gray and the inside is white. There is some gray in that white, but generally speaking, cortex is gray and the inside is white. The surface of this cover or cortex is not flat. There are some depressions or grooves and some elevations. Elevation, depression, elevation, depression. It's waving like this. The elevation is called gyri, or the single is gyrus. The depression are called sulcus, but some of them are deeper and we call it fissures. So elevations is just gyrus. Depression, shallow depression is sulcus, which is the vast majority, and the deeper one are called fissures. It's like this. If I get this and just cut a little bit like this, we call this sulcus. If I cut all the way deep in, we call it fissure. Cerebellum is the second largest after the cerebrum, and this is basically to coordinate body movements. We will discuss the details. This is just an introduction. Just like the cerebrum, it's also two hemispheres, and it is also covered by a cortex that's gray matter as well. Then the diencephalon. The diencephalon is located somewhere in between those two, in between the cerebrum and cerebellum. Basically, there is a thalamus, under the thalamus, above the thalamus. That's basically what it is. Thalamus, above it and under it. Okay? Thalamus is sensory information stop at the thalamus before going further, before going up. She should stop at the thalamus. There is part above the thalamus, we will talk about it, and part under the thalamus, which is called hypo. Epi and hypo. Hypo is under the thalamus. Hypothalamus, it has a lot of functions. Emotions, autonomic functions, hormone production. This is just an introduction again, and these are the broad lines. We will discuss the details. Other parts of the diencephalon, pituitary gland. This is under the influence or under the control of the hypothalamus that I just mentioned. Okay? So what's connecting the hypothalamus? I will show you guys the picture coming. But what's connecting the hypothalamus to this pituitary gland? The other name for this gland is hypophyseal gland. You will see it coming, but when you see hypophysis or hypophyseal gland, it is pituitary gland. It's the more scientific name or the other name for it. So there is a pituitary gland, and what's connecting it to its manager, the hypothalamus, something called infundibulum or stalk. And we have a lot of models in the lab that we will discuss this part. Next part, or the last part here, is called the brain stem. And the brain stem is a stem that's connecting what's above to what's under. Above it and behind it and under it. It's something in the middle, a stem in the middle. Above it, diencephalon and cerebrum. Behind it, cerebellum. Under, spinal cord. And there are three parts for this. From above going down, midbrain, pons, medulla oblongata. Midbrain, pons, medulla oblongata. This is the sequence from above going down. Midbrain, generally speaking, it processes sight, sound, reflexes, maintaining consciousness. This is just generally speaking again. We will discuss the details. It's for processing vision and sound and so on. Underneath the midbrain, there is pons. And this is basically, the pons is connecting the cerebellum to the brainstem. It's behind it is the cerebellum. What do we have in the pons? We have some tracts, relay centers, and some nuclei. Nuclei for what? Nucleus, and generally speaking, do you guys remember the ganglion or the ganglia, which is a collection of cell bodies? The ganglia is a collection of cell bodies outside the central nervous system. So what do you call a collection of cell bodies inside the central nervous system? We call it nucleus. Clear? Collection of cell bodies outside, we call it ganglion. Collection of cell bodies inside, we call it nucleus. So there are nuclei for both somatic and visceral motor control. Last part is the medulla oblongata, and this is connecting anything above it to the spinal cord. Inside of which there will be part of the central canal that we talked about in the spinal cord, if you guys still remember. Do you remember this canal in the middle of the spinal cord in which the CSF is circulating? It will start here. And this is basically the regulation of autonomic functions. Heart, controlling the heart rate and blood pressure and digestion. Okay, look at this picture right here. This part up here is the diencephalon. And then look at those three, green, blue, and like Havan, green, blue, Havan right here. Those three together are the brainstem. Top The top one is midbrain, followed by pons, and the lower one is the medulla oblongata. Okay? What is this diencephalon? This is the thalamus, and underneath it is hypothalamus. And look what's connected to it down here. You see this? What's the diencephalon? Thalamus, hypothalamus, and then connected to the hypothalamus is this gland here. You will see it in the lab. This is called the pituitary gland or hypophyseal gland or hypophysis. Pituitary, hypophysis, hypophyseal, same thing. And you see the connection here, this connection connecting the hypothalamus to the pituitary. This is called the stalk or the infundibulum. Okay, so what you're seeing, and we have a model like this in the lab. What you're seeing here is just two structures. These two structures are diencephalon on top, brainstem at the bottom. So starting with, so this was everything so far is just an introduction to the main parts. One more time, the brain. We finished the spinal cord. Now we're doing the brain. What are the main parts of the brain? Cerebrum, the two hemispheres. Cerebellum in the back. Diencephalon, underneath it, brainstem. Cerebrum, cerebellum, diencephalon, brainstem. These are the main parts. We will start with the cerebrum, the two hemispheres, right and left. First thing is, where do they come from? The embryonic development. And we're not going to go too deep, but you need to know where do they come from, generally speaking. So in embryo, there used to be three vesicles from which all the parts that I talked about will come. Prosencephalon, mesencephalon, and rhombencephalon. You will notice that all of them end with cephalon. Cephalon means brain. There is prosen, mesen, and rhombencephalon. Bras or brasi means front or forebrain. Mesen is middle. It's even sound kind of similar a little bit. Mesen middle or midbrain. Rhombi means behind or the back, hindbrain. These are the three, from which you will have 5 secondary, tele and diencephalon, mesencephalon. Mesencephalon is the same thing, it didn't change. And then metencephalon and myelencephalon. At the end, diencephalon and mesencephalon, they will stay as it is, so that's 2. Telencephalon will make the cerebrum, metencephalon will make cerebellum and pons, and myelencephalon will make the medulla oblongata, and here it is. So this is what you need to know here. This slide right here. Primary, prosen, mesen, rhombi. Mesen doesn't change at all. So this is the easy one. Mesencephalon become what? Become mesencephalon. Secondary. And then it will become still mesencephalon, which is a midbrain. Midbrain. So this doesn't change. What changed are the other two. Prosi, pro, front. Prosencephalon, this is the one that will give you telencephalon that will become cerebrum, diencephalon that will stay as it is. And then the rhombi, the back one or the hind brain, will become metencephalon and myelencephalon. Metencephalon give you cerebellum, pons, and myelencephalon give you the medulla oblongata. This is the origin that you need to know is this slide that I just talked about. Now inside the cerebrum, inside those two hemispheres, we have spaces. It's not like it's compact. There are spaces inside. And these spaces are lined to enclose them. There are different spaces. Why do we have spaces? Because inside the brain, the blood does not enter all the way into the brain. It doesn't happen like this because the brain is too important and too sensitive to allow the blood to enter inside the brain. There is a filter, there is a barrier. The blood should stop. And you should allow only part of it to pass and this part is called cerebrospinal fluid. Cerebro, brain, spinal cord. This is the fluid inside the central nervous system. Are we following so far? So this is why do we have spaces called ventricles? CSF, cerebrospinal fluid. This fluid, why do you have this fluid? Because I can't allow the blood to enter into the brain. Too sensitive, too important. There can be harmful things in the blood. So you have to stop it. There is a barrier. This barrier will allow part of it, water and certain things, including the nutrients and oxygen, certain things are allowed to go in. And these parts that are allowed to go in, CSF. So we make it in the ventricles. Every ventricle will have a part that will filter out and make the CSF. So these are called ventricles. And these ventricles, we have 4 different ventricles. 2 of them are called laterals, and then third and fourth. Look at this picture here. We have a model like this. And there is another one that will show you more. But look at this. This is left. And this is also another left. So this is like right and left laterals. And then this one that have an opening here, this is the third. And last one here is the fourth. And the question that I always hear is, why lateral, third, fourth? Where is first and second? We don't use first and second anymore. We just say right and left laterals instead of first and second. Third and fourth is as is. We didn't change it. So lateral, third, fourth. Right lateral, left lateral, third and fourth. Now what's happening here? I just told you that this is where we make the CSF. So where do we make the CSF? In the laterals first. And then this CSF is going to go to the third. The third will also add its own cerebrospinal fluid. But how the CSF move from laterals to the third? What's the connection there? There is an opening called interventricular foramen. Everything that I'm saying is important, as you guys know by now. I'm not focusing on anything that's not important. So this is how it circulates. This is how we make it and how it circulates. Where do we make it? Lateral ventricles first. And then what? It moved down something called interventricular foramen, the opening that between the ventricles. Which ventricles? Lateral and third. That's where the name came from. Interventricular, enter means in between. The opening that in between the ventricles. So moving to the third. Does the third make its own CSF as well? Yes, it will add its own CSF. And from the third, it go to the fourth. Through what? Through a duct that's called cerebral aqueduct. From the third to the fourth, cerebral aqueduct. Why did you call it this name? Aqua means water or watery fluid. It's a duct that connect or conduct the CSF, which is watery fluid, from the third to the fourth. And it passed through the cerebrum. Going into the fourth ventricle. And then what? And then it go to the central canal, starting from the medulla oblongata. And this central canal is the one that will follow all the way down in the spinal cord. We talked about this. You guys remember this cross-section of the spinal cord? Exactly in the middle, there is an opening. It's not an opening, it's a canal, which is called the central canal of the spinal cord. So this is how it works. Look at this picture here, same thing, but you're looking from the back. Here is right lateral, left lateral, interventricular foramina to the third. From the third, cerebral aqueduct to the fourth, then to the central canal. Okay, how the brain is protected physically? Of course, we have the skull, bones. This part specifically, not without the facial bones. This part is called the cranial bones. So cranial bones, and then cranial meninges. Do you guys remember the spinal meninges? What's the innermost one of these covers? Pia, followed by arachnoid. And the outermost is the durable one, the one that faces the bone, durable. Same thing here. It's the same thing. Same arrangement, same everything, but we call it cranial because this is a brain cover. And also the cerebrospinal fluid. How the cerebrospinal fluid is protecting? Because it acts as a cushion, right? If something happened and you're moving, like a vigorous movement, sudden movement, if you have fluid inside, it will act as a cushion, right? Like you're falling in a water mattress. That will absorb shock, right? So it is in the ventricles and also around the brain. Do you guys remember the subarachnoid space? What was the importance of the subarachnoid space? If you have a question like this. What does it have? No, the subarachnoid, not the epidural. Subarachnoid. What's the importance? I told you one thing important about each one of those. Subarachnoid. What do you have? No. What's the importance? What if this is a question, which will be a question? Of course, this is something important. Cerebrospinal fluid. This is where the cerebrospinal fluid circulates. You guys should remember this. Remember when I told you about the spinal tap? We get a sample. You get it from there, from the subarachnoid space. And what's the significance or importance of the pia? What runs on its surface? Anybody? Blood vessels, yes. And what's the significance of the epidural space? Fat for anesthesia, yes. You guys should remember this. Now this is a physical protection. How about biochemical protection, meaning, or isolation, meaning how chemicals, the harmful chemicals are not allowed in blood-brain barrier, BBB, blood-brain barrier. There is a barrier between the blood and the brain. Starting with this protection. Bones, we talked about it. But how about the meninges? Same thing like the spinal cord. They are continuous with the spinal cord ones. The dura mater, which is the outermost one, is 2 layers. It is 2 layers here. Okay, it is 2 layers. There is an outer one that's actually adherent to the bone, and there is an inner one which is adherent to the or not adherent, I'm sorry, which is part of the meninges. Pia, arachnoid, dura, 2 duras. One is the inner one that's part of the meninges. We call it meningeal layer. And the outer one, we call it the periosteal because it is connected, fused to the periosteum. So what do you have in between those two? Does anybody know? If that's the case. Pia, arachnoid, dura. What's in between those two parts of the dura? What's the space? Epidural. This is the epidural, outside of the dura. Epidural space. Epidural space. Yes, because it's outside one of them. So we call it epidural. Epi means outside. It's outside one of the 2 layers of the dura. Now, this dura mater specifically, the dura mater here is different. It's not exactly like what the one on the spinal cord. The spinal cord, we just said there is a dura mater and there is space outside of it and inside of it. That's it, right? But here the situation is different. The dura mater surrounding the brain, remember the brain is not really like a ball. It's not really like spherical structure. It is 2 halves, right? And these 2 halves, there are spaces in between. Otherwise, how is it 2 halves, right? There is a right hemisphere and left hemisphere. And in between, there is a space. And underneath, there is a space. There are spaces. What do I mean by that? I mean, when the dura mater is covering the brain like this, above it, and then it will find the space. It's going to dig into that space, making a fold. Clear? It's going like this. And there is a space. Yes, make a fold and then continue. Got it? Anytime you see a space, you're going to make a fold. And we call those dural folds, the folds of the dura. Why do we need these folds? Because this is how the CSF is going to circulate. We will talk about the rest of the circulation, but it's making a space just like this. If this is the dura mater and this is the brain is two parts. When it finds a space, it will make a fold like this. Can you see this? This is a fold, and it is not like two layers are not adherent. It's exactly like this. Can you see this? Where you're leaving like a canal here. So there is a canal like this. This is the dural folds. So it can allow CSF to circulate inside of this. So why again are we making these folds? Because these folds will be called the part of which that's going to collect the CSF at the end. We are going to call them venous sinuses. What I showed you, this space in the fold or this canal in the fold, are called venous sinuses, dural venous sinuses. What are these folds exactly? I told you that when you see a space, you make a fold, right? What are these folds? And what are the spaces containing these folds? And this can make a good question. Lab and lecture. Where exactly? Like you're going like this. You found the space. What's the name of the space? And what's the name of the fold inside of which? This can make, again, lab and lecture, both of them. So the first one is called falx cerebri. This will go in the space in between the two hemispheres. This space or fissure is called longitudinal fissure. Next is called tentorium cerebelli. What does it mean? Where did you get that name from? Falx cerebri, the first one. Falx means the separator. Cerebri means cerebrum. This is the separator of the two hemispheres of the cerebrum. Does that make sense? If it doesn't make sense, just let me know. Just say it doesn't make sense. I will explain it again. The cerebrum, the big one, two hemispheres, right? And there is a space in between. So when you go into that space and make a fold, you're separating the two hemispheres. So we call it falx cerebri, the separator of the two halves of the cerebrum. Yes, this is the separator. It separates them. Falx cerebri, the separator of the two hemispheres. No, not the ventricles. These are dural sinuses. This is completely different. The dural sinuses are deep inside the brain. This is surrounding the brain. So we're talking about two different things. Okay? Are you guys following? The first spaces that I talked about, the ventricle, are deep inside the brain, all the way inside. But what I'm talking about now is the cover outside. These are the meninges surrounding the whole brain from outside. This is a space and this is a space. But we're talking about two different things. Next is called tentorium cerebelli. What does it mean? Let me explain it easily. Cerebral hemispheres are up here. All right? Cerebellum is in the back here. So these are two different parts, isn't it? Cerebrum and cerebellum. There is a space in between those two. So if you make a fold in that space, this is called a transverse fissure. So if you make a fold in that space, aren't you covering the cerebellum? Do you guys get what I mean? This is cerebrum and this is cerebellum. Clear? Cerebrum and cerebellum is underneath like this. If you put a fold in between those two, this fold will be covering the cerebellum, right? That's why we call it tentorium cerebelli. What does it mean? The tent of the cerebellum. Isn't it above it like this? Like a tent, right? tentorium cerebelli. I will show you pictures, but this is generally speaking. So this is very important here. Falx cerebri, where is it? In between the two cerebral hemispheres. What do you call it? Longitudinal fissure. You will see it. And since it's a fold, it will allow fluids to move in it, making the sinuses. What are these sinuses? Superior and inferior sagittal sinus. You will see the picture. How about the tentorium? In the transverse fissure between the cerebellum and cerebrum. What is the sinus inside of this? Transverse sinus, because it's transverse fissure. Last one, falx cerebelli. What's a falx cerebelli? Did I explain the falx cerebri? Are you guys following the difference? Cerebrum, cerebellum. Cerebrum, no L's. Cerebellum. Cerebrum, the two hemispheres, the big hemispheres. Cerebellum with two L's, this is the tiny brain in the back. Are we clear? Remember I told you falx cerebri is that separator between the two hemispheres, right? Of what? Of the cerebrum, the big one. How about the separator in between the small one? Same name, but instead of falx cerebri, separator of the two cerebral hemispheres, this is a separator of the two cerebellar hemispheres. Okay? Meninges, same thing. We have pia, arachnoid, dura. Pia is attached to the brain, just like what we mentioned in the spinal cord. And then arachnoid is the middle one, under which you have the subarachnoid space for the CSF. And then there is a subdural space between the dura and the arachnoid. It's exactly the same as we mentioned in the spinal cord. So look at this. Look at this picture right here, and look at this section. This is the cortex covering the brain. It's part of the brain. This is the cover of the brain. And then you have pia adherent. Can we see it here? It's even the same color, the brown here. And then this layer right here, the blue, this is arachnoid. So what is this part here between arachnoid and pia? Subarachnoid space. For what? For CSF. Subarachnoid space. So pia followed by arachnoid. And then do you see those two layers? Both of them have the same name and there is blue in between. This and this, both of them are the dura matter. This is the meningeal one, and this is the periosteal one, and in between the space is called sinus. Is that clear? When you make a fold, or in between the two parts, there is a space in between called the subdural space. And this part, in between those two, we call it sinus. So these are one of the sinuses. Basically, this is what we have here. Look at this picture just to have an idea what are we talking about. This is the cerebrum itself. What's the outermost cover? What's the cover of the cerebrum? Cortex. Outside of the cortex, we have three meninges. Okay? 1, 2, 3. Here it is. Pia, adherent. Arachnoid, not adherent. So there is a space, subarachnoid space. For what? CSF, cerebrospinal fluid. And then what? What's outside of the arachnoid? Dura. And subdural spaces in between, it doesn't have significance that we need to know. Just a name. What's the name of the space between the dura and the arachnoid? Subdural space. That's it. But look at the two layers of the dura again. This is the meningeal one because it's part of the meninges, and this is the periosteal one because it's in contact and adherent to the bone, and in between there is a space, we call it a sinus. Remember when I told you this is the cerebrum? Look at the cerebellum now, it will make more sense. Look at this. This is the cerebrum and this is the cerebellum. Look, if you make a fold here, can you see this area here or this space here? between the cerebrum and cerebellum. Can we see it in the picture? So when you make a fold here inside this, what is this? Transverse fissure. And what do you call this fold? Tentorium cerebelli. Why? Don't you see it's making a tent over the cerebellum? Does it make sense? You're making a tent over the cerebellum. So tentorium cerebelli, inside of which we have the transverse sinus. Now, what is this? This is the falx cerebri. How about this one here? Falx cerebelli. How about this? Do you see this space right here? Can you guys see this space? This space should contain the cerebellum. You don't see it, but it is here. So this is the tent above the cerebellum, and this is a falx in between the two cerebellar hemispheres, and this up here is the falx cerebri, the separator in between the two hemispheres. Now, what are the name of the sinuses running in this? And we have a model that looks exactly like this in the lab. What are the sinuses here? This falx contain one here on top, superior, and one inferior. Look at this. What do you call this plane right here? If I'm cutting here, what do you call it? Sagittal. So if I have two sinuses here, sagittal, one above and one under, one superior, one inferior, what should I call it? Superior, inferior, sagittal sinus. Make sense? Like this. It's in the sagittal, superior and inferior sagittal sinus. What else do we have? We also have continuation of the inferior. It will go straight, like this, straight sinus. And there is one that goes transversely. Transverse is like this, right? These are the basics in the very, very beginning. Sagittal and coronal and transverse and longitudinal. You guys remember this in the very beginning? So if it goes like this, it's sagittal. If it goes to the sides, it's transverse. Okay, so you do need to know these sinuses. So superior sagittal sinus, inferior sagittal sinus, straight sinus, transverse sinus, and this one down here, it's not labeled, but it looks like sigma, so we call it sigmoid sinus. Here is the cerebrospinal fluid I was talking about. Where do we make the CSF, cerebrospinal fluid? Ventricles, inside the ventricles. How many ventricles do we have? 4, right? So why do we have CSF? Number one, cushion, act as a cushion, right? Like a water mattress. If you have fluids, it will act as a cushion. This is one. Number two, it's also support. Number three, why do we have it in the first place? Because we cannot allow the blood in, right? So what was the function of the blood? What's the main function of the blood? Isn't it to transport? The blood is transport. What else is the function? Transport. Oxygen, nutrients, and carbon dioxide, isn't it? Why do we have blood? To give you nutrients and oxygen, and to take away waste products and carbon dioxide. Isn't this is the basic function of the blood? Same thing. But the blood is not allowed in. CSF will do that work. So it is transport. Transporting what? Oxygen and nutrients to the brain tissues and taking away waste products and carbon dioxide. It also transmits chemical messengers. So I've been saying that we make the CSF in the ventricles. We make the CSF in the ventricles. Yes. What exactly make it in the ventricles? Choroid plexus, something called choroid plexus. Choroid plexus, capillaries surrounded by ependymal cells. Do you guys remember this or not? I told you about these cells, and I told you question in each one. Ependymal cells, remember when I told you CSF? Here it is, ependymal cells. Surrounding the capillaries, we call it choroid plexus. This is where we make the CSF. About half a liter per day in all these ventricles. 500 milliliters per day. So choroid plexus is going to make the CSF, make it and remove it and adjust its composition. Now, how the CSF circulates? We know CSF will make it in the ventricles, right? What exactly make it in the ventricles? Choroid plexus, right? So choroid plexus make it in the lateral ventricles. It moved from the lateral ventricles to the third through interventricular foramen. From the third, it has its own choroid plexus. It will add more, right? Now I have more CSF. Where does it go? To the fourth. Through what? Aqueduct. The fourth will add its own because it has its own choroid plexuses. Now we have a lot of CSF in the fourth ventricle. It will leave through the central canal of the spinal cord, moving all the way, central canal, central canal, central canal. At the end, it's going to go to the subarachnoid space and come back up. Are we following? Like this. Lateral ventricles to the third, to the fourth, to the central canal. Leaving the central canal, go to the subarachnoid space. Remember I told you subarachnoid space where you get sample? Subarachnoid space. Subarachnoid space of what? Of the spinal cord. Then it will move up to the subarachnoid space of the brain. Then we will take it out of there. To what? To the sinuses. And from the sinuses, ultimately, it will go back to the veins, back to the heart. Can you imagine the whole circulation? Let me just wrap it up together and help you understand. What are we trying to make here? We're trying to make CSF, fluid inside the brain, because blood cannot go in. How to solve this issue? I cannot allow the blood deep into the brain. No. So what should we do now? Arteries going to the brain, branching, branching, branching until the capillaries. But the capillaries contain blood, right? Can you allow the blood to go into the brain? No. The capillaries with ependymal cells, choroid plexus, inside the ventricles, we will allow the fluid part that's filtered, what we need only, selective, to go into the ventricles. Lateral, third, adding more, choroid plexus adding more. Fourth, choroid plexus adding more. Are we following? What's after the fourth? Central canal, moving all the way down. Clear? What's after the central canal? It will leave the central canal, go to the subarachnoid space. Right? This is the spinal cord surrounded by layers, right? It will leave and go to the subarachnoid space. And then what? To the subarachnoid space around the brain. Following? And there is a structure, we'll take it from there, pour it into the sinuses. And the sinuses will take it out back to the veins, back to the heart. So you started from the heart, you ended in the heart. Clear? From the heart to the arteries, to the choroid plexus, CSF, circulating around. At the end, you take it out of the subarachnoid space by something called arachnoid granulations, or villi, you will see it, and pour it into the sinuses. In the sinuses, it was going to move down the sinuses, going to the internal jugular vein that will return it back to the right side of the heart. So you got oxygenated blood with nutrients, and you leave deoxygenated going back to the heart, and the heart will do the job, and you keep circulating 24/7 just like this. Okay? So I mentioned that, and I will show you the pictures. I mentioned that after all this circulation, you end up going to the subarachnoid space of the brain. Is that clear? This end part is clear. So now I'm in our subarachnoid space. Now what is this? This is the last station, the last part. We already did everything. We delivered what we need to deliver, oxygen, nutrients, and we're taking waste products. Now it's time to leave. How to leave? Arachnoid villi, arachnoid granulations. These are the ones that are going to absorb CSF from the subarachnoid space and send it to the sinuses. Look at this picture here. So lateral ventricles, third ventricles, moving down, fourth ventricles. Look, central canal of the spinal cord, subarachnoid space is number 4, and then it will go to the subarachnoid space of the brain itself, which is this green right here. Can you see this? The red arrows, from which these arachnoid granulations and villi will take it and pour it into this, the blue. And the blue are the sinuses. And look what's happening here. Superior sagittal sinus, inferior sagittal sinus, straight, transverse, and then it will leave through the internal jugular vein. Do you guys remember the jugular foramen in the skull? This is where the internal jugular vein leave and go to the heart. So this is the choroid plexus, capillaries surrounded by ependymal cells. It's going to allow part of it to leave out. Capillaries surrounded by the ependymal cells, and the ependymal cells are going to allow certain parts to leave and go to the ventricles. Look at this. These are the subarachnoid, the arachnoid granulations and villi. We'll take it from here. What is this? Subarachnoid space into the sinuses. So we talked about the protection of the brain, including the CSF part and the blood supply of the brain. Blood supply of the brain. Remember I told you, the blood goes to the brain. Does it leave the capillaries and go into the brain tissues? No. But how did it reach the brain in the first place? Like we are here in the heart. How does it goes all the way to the brain? Two routes or two pathways: internal carotid artery and vertebral arteries. 4, internal carotid here in the front and vertebral in the back. Do you guys remember the vertebral foramina? In which vertebra? Cervical or thoracic or lumbar? Cervical. Do you remember these openings? This is where this artery goes. It goes in these. The transverse foramina, if you remember, this is where the vertebral artery goes into these foramina until it enter into the brain. This is one. Here in the front, do you know when we like put our fingers like this to pulsate, right? To pulsate and feel the pulse here. Like if we do like this, I don't feel something here. This is the most common location right here. Okay, the radial artery. If you wanted to feel the pulse, usually we use this. 3 fingers like this and we feel it. There is a pulse here. But what if somebody is like comatose or unconscious or something and you don't feel the pulse here? We usually feel here. If there is no pulse here, he's almost dead because this is very close to the heart and it's very big. What is this? Carotid. Internal one go to the brain. So what are the 4 sources? 2 internal carotid in the front, 2 vertebral in the back. And we have models for these. So obviously this blood circulation should continue 24/7. If something happened that interfere, those patients will get into stroke. And I think we're all familiar with this. If you stop the blood going to the brain somehow, it will lead to stroke, right? Which is called cerebrovascular accident. But does that happen if the blood goes less? No. Shut down. If the blood shut down, even for few seconds, but complete shutdown, not partial, not less, it will lead to cerebrovascular accident, also known as stroke. And stroke, the neurons will actually die, not just deprived, die. Cerebrovascular accident. Now the blood-brain barrier that I talked about, which is the isolation between the general circulation of the blood from the CNS. This barrier is made by a tight junction, which is a very close junction. The cells come very close to each other between the capillary endothelium. The endothelial cells of the capillaries, they come very close to each other to the extent of making tight junctions. Astrocytes are going to regulate this barrier by releasing chemicals to control it. It tells the barrier, allow more, allow less, astrocytes. And what can pass through this is certain things. It's selective. You have a barrier, but certain things are allowed to go through it. These things are selective. You select what goes and what doesn't go. Generally speaking, oxygen and carbon dioxide is allowed, obviously, because you want oxygen to go to the brain and you want carbon dioxide to leave. So this easily just pass in and out. Steroids, prostaglandins also can pass this. So just remember certain things, including oxygen and carbon dioxide and steroids. Blood CSF barrier, this is another component that's helping out, and this is basically the ependymal cells. Okay, so what do we have here? We have blood-brain barrier and blood CSF barrier. Blood-brain barrier. This is the part with the capillaries. What's surrounding the capillaries in this area to make the choroid plexus? Ependymal cells. So you have two components here, blood-brain barrier and blood CSF barrier. Blood-brain barrier, endothelial cells connected by tight junctions. Are we following? How about the blood CSF barrier? Ependymal cells also connected by tight junction. And this is the part that's going to together make the choroid plexus that's allow the CSF or making the CSF. Is that blood-brain barrier completely separating like a complete spherical structure? No, you cannot do that. You should allow the brain to communicate with some structures outside. So it's not like a complete spherical structure, right? It's not a complete cover. No, there are certain breaks to allow communication, and you do need to know these breaks. One of them is for the hypothalamus. The hypothalamus should communicate. So how does it release its contents? You have to have a break for the hypothalamus. Another one for the posterior lobe of the pituitary gland. Posterior pituitary gland. Because this is where we store the hormones, ADH and oxytocin, so you should allow it to communicate. You also have a break for the pineal gland so that it releases its secretions. And the last one is for the choroid plexus. This one also have its break to allow the CSF, to maintain the CSF. Okay, we are going to go to the different parts of the brainstem, but so now we're getting into the different parts of the brainstem. The first one is the medulla oblongata, which is the lowest one, the one at the bottom. And this one, let me say it this way, just to make it easy for you. Listen, each one of the three parts of the brainstem, the number one function is the same in all of them. It's all about location. Like this is the midbrain. Where is it located? Under the diencephalon. So what's the function? Connect the diencephalon to the pons. That's it. How about the pons? The pons is in the middle. What does it do? Connect those two parts. Okay? So the pons connect midbrain to the medulla. What's the function of the medulla? Connecting the pons above it to the spinal cord underneath it, right? So the first function is function by location. The function by location. If she's sitting down here, her function is to connect them. That's it. This is the first one. Her function is to connect them, and so on. So if you know what is the location, connecting. That's number one. Number two, it contains, this is the one that I just mentioned here. It's a relay station or communication between the brain and the spinal cord because it's the lowermost one. And this goes without saying. If you know the location, you know the first function. But it is controlling the visceral functions, containing nuclei for the visceral functions. And coordination of the autonomic reflexes. Visceral functions, controlling visceral functions, and reflexes, autonomic reflexes. If you look at this picture again, we saw this before. If you guys remember, up here is the diencephalon. And these three parts, green, blue, orange, or Havan. What is this green on top here? This is the midbrain. What is this blue in the middle? Pons. What is the most part? This is the medulla, after which you have the spinal cord. But the three parts are, from above going down, midbrain, pons, medulla oblongata. First function is location. Second function, cranial nerves come from this part. Which cranial nerve come from each one of the three? Third function, nuclei inside. One more time. What's the brain stem? Three parts. Midbrain on top, pons in the middle, medulla at the bottom. Is that clear? Three parts. What's the function? First function, location. Where are you? Where are you? Where are you? Connecting. Clear? This is number one. Are we following? Number one is location. What's number two? Cranial nerves come from this. Cranial nerves come from this. Cranial nerves come from this. We will learn it and I will put it together to remind you. Number three, what kind of nuclei do we have? I talked about the medulla oblongata, reflexes and autonomic functions, like respiratory rates, like digestive function, like cardiovascular function, and so on. So we will get it one by one. But look at this here. Look at this. You will see that 3 and 4 coming from the midbrain. 5, 6, 7, and 8 come from the pons, and the rest come from the medulla oblongata. You got one more function here. First function is location, connecting. Second function, cranial nerves. Third function, which is harder, is the nuclei. So first function is simply, what is the function of midbrain here? Where are you? I'm in between the thalamus and hypothalamus above, which is the diencephalon and pons. That's it. You're connecting the diencephalon to the pons. What's the function of the pons? Connecting the midbrain above it to the medulla oblongata, underneath it, and the cerebellum behind it. How about the medulla? Connecting the pons to the spinal cord. I think it's straightforward, just location. How about the second function? Second function is 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 2, 4, 4. So how about cranial nerve 1 and 2? No, this is something else. It's not related to those. So 1 and 2 by themselves. We will talk about it. But the rest, the other 10, are arranged in 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Here it is again. Oh, of course, yes. For the lecture and the lab. Where is 3 and 4? Right here. 3 and 4. Midbrain, the green. 5, 6, 7, 8. Here is 5, 6, 7, 8. Pons. And then 9, 11, 12, 9, 10, 11, and 12 is all by itself here. Where are these located? Medulla oblongata. That's it. I think that's easy. 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 2, 4, 4. This is again up here is the thalamus. You guys see the choroid plexuses? This is how it looks like. You see this red? choroid plexus in all locations. Lateral and third and fourth. This is where it's located. But I want you to look at this here. Can you see this section? What is this part? If you guys are following, what is this? The pons, right? You see what's connected to the pons here? 1, 2, 3. There are 3 things connecting here, if you can see it. These are called cerebellar peduncles. Cerebellum, cerebellum. Do you remember the cerebellum in the back? The small one? Peduncle means like a bridge, like a connection. So there are 3 connections connecting the cerebellum behind to the pons in front of it. Look at this picture here. This is the midbrain, this is the pons, and this is the medulla at the bottom. Midbrain, pons, medulla oblongata. Look at the pons, superior, middle, and inferior peduncles. This is obviously the 4th ventricle here. What are these autonomic reflexes that I was talking about? Reticular formation, cardiovascular centers, and respiratory rhythmicity centers. You do need to know this. So if we talk about the medulla oblongata, first function is location, connection. Second function is the last 4 cranial nerves. Third function, what are your nuclei doing? What are you doing here? Reticular formation, sleep, wake, and controlling the autonomic function. Cardiovascular, the name gave it away. Cardiovascular, controlling the heart and the blood vessels. Respiratory rhythmicity, what does it mean? You know what the rhythm means? You go in fixed interval, 1, 2, 3, 4, like 1 per second, or 1 every 5 seconds, for example. This is called rhythm, right? So like if you're breathing, you go like 1, 2, 3. It should be almost the same sequence. Like if you're sleeping, do you go like irregular? If somebody's watching you and you're breathing, you're sleeping, it's not irregular. You do like this. You go in fixed intervals. These are called rhythmicity. You go in a rhythm. And this rhythmicity is decided by the medulla oblongata. And understand this because the pons, the next one, also control the respiratory system, but in a different way. So let me say this way. If you count your respiratory rate, if I count my respiratory rate, and I'm just resting like this, count your 20, let's say 20, okay? So who decided the 20? The medulla oblongata. Does it always stay 20? No. What if I'm exercising? What if I'm walking, running? Is it going to change? This is the pons, not the medulla. So the medulla is rhythmicity. It set the rhythm. Pons control the rhythm, regulate the rhythm. So it changes it like its supervisor, something like this. What kind of nerves do we have here? The last 4 and part of 8. 8 is in common between those two, okay? So 8, 9, 10, 11, 12. I usually do it like this. This is something, of course, that you have to know. Here and in the cranial nerves and in the lab. I will do the, like, the midbrain will be something like this, followed by the pons, followed by the medulla, and then the spinal cord, right? 3, 4, 5, 6, 7, 8. 8, 9, 10, 11, 12. What do you mean? I mean 8 come from both of them. Is this clear? You have to know this. This will give you a couple of questions just by knowing this. What else do you have in the medulla oblongata? I have 2 nuclei, gracilis and cuneatus. These are the ones that are controlling or in which something called pyramids are passing to control the movements. There is also solitary nucleus for visceral sensation, inferior olive. This is for the somatic motor commands. But what I told you is the most important ones. Respiratory and cardiovascular system. This is the most important ones. So here it is. Look at this. These are the pyramids. It's part of it. This is the inferior olive right here on the sides. Pyramids and olive. And right here, these different parts here are reticular formation, cardiovascular center, solitary nucleus. One more time, the most important thing that you have to remember, respiratory rhythmicity and cardiovascular system. There are other functions, but you cannot forget those two. Most important two. How about the pons? The pons will contain some cranial nerves, and part of it is for location. Function number 1 and 2, it goes without saying. What cranial nerves do you have here? It's right here. 5, 6, 7, 8. And location, yes, between the midbrain and the medulla, and behind it is the cerebellum. Like this. 3 peduncles, 1, 2, 3, and the cerebellum is like this. Function by location. And what else? Superior, middle, and inferior. Yes. And what do we have here? The most important, the bottom line that you cannot forget. Respiratory centers called apneustic and pneumotaxic centers. What are these? These are the ones that modulate. Medulla is setting the pace. 20 per minute, 18 per minute, 15 per minute, and that's it. Just making the rhythm. Pons above it, modify it, change it. Go higher, go less. Go up, go down. Breathe more, breathe less. Those two centers. Otherwise, other functions, okay, but you cannot forget this one. Midbrain. Midbrain is two parts itself. One of them is called tectum, and the other one is called tegmentum. Both of them start with T. Tectum, tegmentum. Tectum, tegmentum. Together, midbrain. What's a tectum? Tectum is you have 4 bodies like this. You see this? 4 bodies, 2 in the left and 2 in the right. 4 bodies. 4 means quad, right? Do you know this? Bi, tri, quad. So quadriceps. Quadrigemina. Corpora quadrigemina. Quad means 4. You have the 4 bodies. You will see it in the lab. 4 bodies. 2 superior right here, right and left, and 2 inferior. Clear? This is part of the midbrain. Superior is for vision, and the inferior is for auditory function. You need to know this. The 2 parts of the midbrain are tectum and tegmentum. What's a tectum? The 4 nuclei. 2 superior, 2 inferior. Superior are for vision, inferior for auditory or hearing. How about the tegmentum? Red nucleus for the blood vessel, substantia nigra. This is to control its gray matter, but it's control the coordination of the muscles. What else is in the midbrain? Cerebral peduncles. You mean the cerebellar peduncles? The 3 superior? No, these are the cerebellar peduncles. Are we following? Cerebellum is in the back, connected to the pons by 3, right? Superior, middle, inferior. How about the midbrain? Connecting above cerebral peduncles. And these are the ones that contain the tracts going up and down. Look at these. Can you see this green? Look in the back here of the midbrain. Can you see these 4? 2 superior and 2 inferior. Together, all 4, corpora quadrigemina. Quad, 4. The 2 superior and the 2 inferior. 2 superior for vision and 2 inferior for auditory function. What do you call each one of those? Colliculus. Corpora quad. Quad what? 4 nuclei. What do you call these nuclei? Colliculus. Superior colliculus, inferior colliculus. Superior is for vision, inferior, auditory function. And then the cerebral peduncles connecting it to the brain. Cerebellum, just like the cerebrum, there is outside cortex and there is inside filling. Which one is gray and which one is white? Cortex, is that gray or white? Gray, and the inside is white. So cerebellar cortex is gray, and it also contains folds that are called folia, and there are two lobes only, anterior and posterior lobe. Is it also two hemispheres? Yes. Cerebrum is two hemispheres, right and left. Cerebellum is two hemispheres as well. Connecting them is something called vermis. Vermis. Vermis means worm because it looks like it. I will show you the pictures. It looks like a worm. Inside of it, there is a tree-like structure called arbor vitae, or the tree of life, arbor vitae, and I will show you it actually looks like this. Most important thing is the nuclei that we have here. And I usually do it like this. This is what I used to do, like when I was in the medical school. I always say to myself, just remember about the cerebellum, BPM. These three letters, cerebellum. If you ask me cerebellum, BPM. B, balance. B, position. M, muscle movement. Enough for the cerebellum. If this is the only thing that you remember about the cerebellum, I'm talking about physiology now. Function. Enough. Balance, position, muscle movement. So once you hear cerebellum, BPM. BPM. Position, balance, position, muscle movements. BPM. Enough. Okay? Anatomy is something else. Yeah, there is a cortex and a vermis and other parts. But for the physiology, enough. Cerebellar peduncles, I talked about it already. This is, the cerebellum is in the back of the brainstem. How is it connected? Three peduncles, superior, middle, and inferior peduncles. If somebody's having a disorder or an issue with the cerebellum, what's going to happen? What do you think? If there is an organ responsible for balance, position, and muscle movement, and this is injured, what do you think? You're losing those. That's it, right? You're losing your balance and your muscle control, isn't it? If you have a function, you lose a function. That's it. But this has a name that I want you to remember, which is ataxia. A means no. Taxia means like balance. Ataxia, no balance and no muscle coordination. Why or details? No, that's good enough. You're responsible for balance and muscle coordination. You have an issue, you lose those two. That's it. We call it ataxia. So look at this. This is the cover outside, which is the cerebellar cortex, anterior lobe, posterior lobe. Look at this worm-like structure. Worm in Greek is called vermis. So this is the vermis. This is what you see here is the cortex. Same thing. Look at this inside. Look at the white matter. Doesn't it look like a tree? Look. The inside. Look at the white inside. You see how it's branching like a tree? This is how the tree, the arbor vitae, or the tree of life, this is how it looks like. So what's the arbor vitae? The white matter, looking like a tree inside, which contain the nuclei. Next is the diencephalon. What's the diencephalon? Thalamus, above it and under it. Thalamus, above it and under it. Thalamus, epithalamus, hypothalamus. Epi above, hypo is under. Okay, what are the different parts? thalamus, epithalamus, hypothalamus. Pineal gland is part of this system above it. And the pineal gland, you need to know that it secretes melatonin. Did we hear about melatonin? The one that help you sleep well and fight aging and all that stuff. Yes, this is where we make it, pineal gland. And this is what you need to remember. Pineal gland, melatonin. How about the thalamus? The thalamus, I call it the secretary of the brain. Of course, this is not a scientific function. We should understand more, but just put this in your mind. This is my secretary of the brain. And imagine what do you think if you go to an office, to the manager's office, and you see the secretary first. Are you just go into the manager, or you have to talk to the secretary first, right? So you go to the secretary. I'm here to bring this information for the manager. Okay, thank you. I'll give it to him. Or I will see what I can do, right? Why are you here? I want to know what time you close and open. Why do you need the manager, right? We open from 9 to 5. That's it, right? So not everything nothing bypass the secretary and go to the brain. The secretary will take the information and let me know what you need. And I will decide what go to the manager and what does not. Why would you go to the manager and say, somebody's asking what time we open? How is that important, right? So the thalamus is the gateway or the entrance to the brain itself. It will decide what goes and what should be stopped. Okay. So what's the function here? It's a relay station for the sensory information, and it decides what's allowed to go to the brain, to the cerebrum and cortex, and what's not. So basically, let me say something like this. If you step on a nail or something, right? Isn't that something that you have to take care of right away? You have to be aware of it, right? The thalamus will say, of course, go to the manager. We have to take care of it right away, right? Immediately. You have to do something, otherwise you'll be bleeding. So this is sharp pain. Yes, the thalamus will allow to go. But compare this to a continuous nagging pain coming from your knee or from your abdomen. Do you think the thalamus will allow to go to the brain? You know what will happen then? You will never fall asleep. If you have a little bit of continuous pain in your knee, don't you just ignore it and sleep? Why? The thalamus block it. The thalamus will say, what's so important to tell the brain, the manager, about something like this? It's not something sharp. It's not something that need immediate attention. So no, it will be blocked. This is what I mean by secretary. It relay the sensation, allow things, and prevent things from going. And the two thalami, there is right one and left one, just like everything else. These are connected by something called interthalamic adhesion. You will see it in the lab. How about the nuclei inside the thalamus? Nuclei. We have 5 nuclei. The anterior one is for the limbic system, which is emotion. It's part of the limbic system. The medial one is for emotions, again, but it's awareness. The ventral one is the one that we are talking about, the most important one, which is relay station for the sensory information. This is the ventral nucleus. Dorsal one, and then there is geniculate one. Lateral is for vision, medial is for auditory, just like the colliculi. Superior and inferior colliculi, the corpora quadrigemina. Remember, superior and inferior, vision and auditory. Here it's lateral and medial. This is the location right here of the thalamus. These are the different parts, but I told you what's the most important of all. Remember that the ventral one is the one that acts as a relay station, and this is the most important one. Yeah, anterior is part of the limbic system, medial is for sensation, integration, and so on, But the bottom line is what you cannot forget is the ventral nucleus relaying sensation going up to uh, the brain. Also, the medial and lateral geniculate bodies. The medial is for auditory and the lateral for vision. More than enough. Hypothalamus. Hypothalamus. Remember, we're talking about thalamus, under and above, right? Thalamus, we talked about it. And more details, don't worry much about it unless you are interested. But I told you the bottom line. This is what you have to know. How about what's under it? The hypothalamus. Hypothalamus is an extremely important part of the body because it contains different things that are controlling like the whole autonomic nervous system. So there is mammillary bodies, and this is contain the reflex for eating. There is infundibulum connecting it to the pituitary underneath. I showed you the picture, remember? There was pituitary gland hanging. This is what's connecting it, infundibulum or the stalk. And then the last part is called the tuber cinerarium. And this is the one that's responsible for the hormones. We have a model that looks like this. This is the thalamus in the middle. And these are the different parts. We will talk about it. This is called corpus callosum, connecting right and left. And there are different parts that we will talk about, but I just want you to see here thalamus. Under it, look at the hypothalamus. And look at this, the infundibulum. They cut it here, but the pituitary gland should be underneath. Here it is again. Look at this. Hypothalamus is up here. You see this hanging? Pituitary gland. And what's the connection? Infundibulum or stalk. What's the other name for the pituitary gland? Anybody? What's the other name? Start with H. Hypophysis. Hypophysis. So what are the nuclei and what is the most important thing about the hypothalamus? I'm giving you the bottom line in each one of those. Okay, cerebellum, BPM. This is the bottom line, right? Thalamus, most important relay station for sensation. Plus the geniculate bodies. There are many other things, yes, but this is what you cannot forget. The bottom line. Okay, so what's the bottom line for the hypothalamus? This slide. Okay, number one, hypothalamus is literally the manager of the endocrine system. Do we know the endocrine system? Endocrine system that make the hormones and control the body. This is the manager, the head. Also, the head of the autonomic nervous system. It's a head of two parts, two systems, endocrine system and autonomic nervous system. Is that clear? This is the manager of both of those. Being the manager or the head of the endocrine system and being part of the nervous system, we're talking about it here, right? So it's a link between those two. It's a link between the nervous system and endocrine system. Are we good so far? Hypothalamus, the head of those two, endocrine and autonomic, and being the head of endocrine, being part of the central nervous system, it's a link between those two. Okay, so when you say the heads of the endocrine system, what does it mean? It means it manages the whole endocrine system. How? By releasing hormones, factors that go to the different glands and tell them what to do. Clear? I'm the manager. I'm the hypothalamus. I manage the endocrine system. How? I make factors, orders to tell every gland what to do. Simply. Okay? All right. Is that it? Like the hypothalamus is sitting down, giving orders to the endocrine glands, you secrete this, you secrete this. No, no. It set an example. It will say, okay, I'm going to make two hormones as well. I'm not just giving the endocrine system orders. Are we following what I mean? If you are the head of something, yes, you're telling everybody, you release your hormones, you release your hormones, you stop, you increase more. You're the manager, right? But you should help a little bit. So I'm going to do some work by myself. What is it? Secreting two things, ADH and oxytocin. This is what the hypothalamus itself make. ADH will come from a nucleus that's called supraoptic nucleus, and you need to remember that. Oxytocin comes from the paraventricular nucleus. So what are the secretions and where does it come from? Important. Hypothalamus, are you only managing the endocrine system? No, I'm helping as well. I secrete those two. The way I remember it, if you're interested, I call it the sad pox. I know this is weird. This is what I use. Okay. Supraoptic, ADH. Paraventricular, oxytocin. Sad pox. If you wanted to use something else, it's up to you. But this is how I remember it. The sad pox. Supraoptic, ADH. Paraventricular, oxytocin. Okay? It also regulates body temperature through something called preoptic area. Do you remember when we talked about the homeostasis in the very beginning? You guys remember this? Homeostasis, that was like the very first chapter 1, homeostasis. Remember that when it gets cold, we heat up, and when it gets hot, we cool it down, and there is a thermostat. Here it is. It's in the hypothalamus. What else? Circadian rhythm by the suprachiasmatic nucleus. Circadian rhythm. I think we know the circadian rhythm, right? If you travel abroad somewhere and you see it's the night and day cycle is disrupted, we call that circadian rhythm. It's controlled by the hypothalamus as well. Feed hunger, thirst, and satiety centers. Hunger and satiety plus thirsty. Food, eating and drinking. It controls eating and drinking. Hunger and satiety and thirst. Limbic system is the next system, and the bottom line is here. Limbic contain M, and M remind me with emotion and memory. Is that enough? Yes, enough. This is what I remember from medical school. Okay? When you tell me limbic system, M, click, M, limbic, M, memory, emotion. M, memory, emotion. That's it. Enough? Yes. Just like the BPM. Okay? If you guys want to use my tricks or shortcuts, use it. If you have your own way, use your own way. But I'm telling you what I used to do, and I still remember it back then until now. I still remember it. When you say cerebellum, BPM, just like this. Limbic system, M, memory, emotion. Done. Good enough. Components of the limbic system: cingulate gyrus. You will see it. Dentate gyrus, another one. Parahippocampal gyrus, hippocampus. It's okay if you remember it, but you cannot forget those two M's. This is the bottom line. And there is another thing that's called amygdaloid body, and this is between the limbic system and the cerebrum. Fornix is also part of the limbic system, plus the anterior nucleus and reticular formation. All these are parts of the limbic system. Here is the limbic system with the different components. Anterior thalamic nucleus, uh, the fornix, cingulate gyrus, parahippocampal, and so on. Okay, cerebrum, and I'm going to stop here at the cerebrum.